And so we're going to go to a third dimension here with Luisa, where basically you were a master's originally and then came on into the asylum and have been there for the last 8 years working on this sort of platform of organoids. Is that correct?
Yeah, I started in my undergrad.
Wow. And never left. And now is developing not just 2-dimension, but 3-dimension and taking it into space as well.
Yeah.
Okay, very cool. Okay. Anyways, welcome. Thank you for, okay.
Okay. So, hi everyone. My name is Luisa Coelho. I am a research associate currently at the University of California in San Diego. And today I will be discussing the use of cortical organoids as a model to study the DLG4 synaptopathy. Just the green button?
Hit the green button.
Cool.
And the pointer doesn't work.
Right. So, first and foremost, I am not a geneticist or a molecular biologist. So I have been so grateful to have you all give me a class about all the ways in which this protein acts in the synaptic terminal and all the isoforms that are generated, and I love to learn about all this, but it's not my focus, so I'm not going to focus on that part. I'll give a very brief background. So, as you all know, the DLG4 gene encodes for the postsynaptic density protein called the PSD95. The PSD95 acts at the postsynaptic density, right, at the postsynaptic terminal here, so not on the presynaptic, on the postsynaptic. Therefore, it is found on the dendrites mostly and not on the axons of the neuron, right? So the PSD95 regulates glutamatergic receptors such as NMDA and AMPA. So it alters dendritic morphology, and it is very important in synaptic maturation and plasticity. So when we have disruptions in the DLG4 gene, we can have the Shine syndrome, right, which disrupts the expression of PSD95. In my case here, I have been working with 4 different mutations. I have a frame shift mutation, I have a splice site mutation, frame shift, but in the splice site region, a nonsense mutation, and a missense mutation, and all of their respective controls. And this is what I'll be discussing today. So first of all, let's talk about organoids and how we generate them and why we use them and why they are beneficial in this case. So the first thing that happens is we receive the skin biopsies from you guys, right? And we're very thankful that you guys offered this to us because it allows this type of work to happen. So from these skin biopsies, we get fibroblasts. And these fibroblasts, we expand them in the tissue culture room, and once we have enough, they go through a reprogramming process. And what that means is that we introduce some key transcription factors through some viral vectors, and we return those fibroblasts we make them go back to being stem cells. We return them to a pluripotent state. So once we have these pluripotent stem cells, they can basically become any tissue of the human body. And so once we have those, over here in this image, the first thing we do is that we verify the pluripotency state of these cells. So on that first image here, we have SOX2 and OCT4 positive cells. These are 2 of these key transcription factors that makes these cells stem, right? Makes them pluripotent. So once we guarantee that our colonies are stem cells, really, now we can start the differentiation process. We induce these stem cells to become neurons, and not just neurons in our case. We actually induce them to recreate the neurodevelopmental process. So they will basically restart the brain development. So what we do, in non-technical terms, we dissociate these colonies. We turn them into single cells, and once they're single cells, we put them on this rotational shaker, make them shake, and they will by themselves aggregate in a 3D structure and form brain organoids. Now, this is a 1-month-long process. From stem cells to brain organoids. It's a 1-month process, and before it's even longer. So you guys can imagine how long it's been taking us to do this, all of this thing with all of the mutations that we received. So our protocol includes 3 different phases. The first one is the neural induction, the second one is the neurodifferentiation, and the third one is the neuromaturation. So these organoids, it takes 1 month to generate them, but once they are 1 month old, they keep growing and they keep developing. And so right now, actually, in the lab, I just left organoids there. They're 1 year old. And so they are very plastic in that sense. So here I have some images to show you guys really how this works. So on the left, we have a 1 month old organoid. So this organoid, you see that it has structures, right? And this first structure here that you see, it looks a little bit like a hole. That's called the neural rosette. So these neural rosettes, this is the beginning of the formation of the neural tube. This is a process that happens during neurodevelopment in the embryonic stage, in the fetus, right? So these rosettes, we induce the formation of them, but once they are there, they will evolve by themselves, spontaneous. They show up by themselves, and what I can tell you about them, so in this first image, in green, we have SOX2 positive cells. You see that these green dots are mostly what you have inside of the neural rosettes, right? So SOX2 is a marker for these stem cells. So inside the rosette, mostly what we have are these stem cells. And then from the stem cells, they will start migrating outwards and differentiating. So in purple, we have nestin, which is a marker for neuroprogenitor cells, and that's what they are becoming. And then in red, we have PAX6, which is a cortical progenitor cell. It's a marker for a cortical progenitor cell. So in that, and you can see that the red is already on the outskirts. It's outside of the neural rosette, which means that they're assuming that cortical identity later in the process. And then here on the right side, this is a 3-month-old organoid. So you can see that this is a completely different organoid. It doesn't have those structures anymore. Those structures are already gone. And now we have a structure that already has a more cortical identity. So in green, first we have TBR1. This is a marker for excitatory synapse, for excitatory neurons, for glutamatergic neurons. And these neurons usually lie on layer 6 of the cortex. So this is very specific, but it shows how the amount of specificity that we can get with this model. CTP2 is a deep layer marker. So these neurons will be found in layer 5 of the organoid, so in deeper layers. And then we have beta-3 tubulin, which is just a marker for differentiated neurons too, and it stands for both axons and dendrites. So this is just to show you how complex these models can be and how plastic they can be. And another really big advantage of using them in studies like DLG4 is because first, they're easily reproducible, right? Not easily, it takes a very long time. But I can make them as many as I need without having to bother any of you, right? The second is just how it's a personalized way of model what is going on in the brain of your kids without invading their bodies in any way, right? I'm basically recreating the neurodevelopment that happened outside of their bodies. And then the third is, I forgot the third. Let's move on. Right. Okay, so what did we do with these organoids, right? We took them and we analyzed them. Oh, right, of course. It's how plastic they are. They change and they continue evolving, which is what I mentioned to you before. So in our case, we know that PSD95 will only, remember, PSD95 is important for synaptic maturation, and therefore we know that they will only start appearing in our organoids at 2 or 3 months of age. So the beginning of this experiment, this study, has been to just grow these organoids, make sure that they have this cortical identity, and wait until they start expressing PSD95 spontaneously, right? So at 3 months of age, we generated cortical organoids from all of these lines and their respective controls, and then what happens is that we collect them. We collect them, we freeze them, we cut them into these very thin slices, and once we have that, we stain them. And Sharon gave a really nice explanation of how this staining occurs with these antibodies, and that's the same thing that we're doing here. And also, And also everyone else here is doing the same thing. So basically what we do is that we get these thin slices of the organoids. We have the proteins PSD95 is expressed throughout in that slice. And then what we do, we add an antibody that is specific to the PSD95, and then we add another antibody that will shine that will glow in a specific color. And then we can see that. So in green here, that's PSD95. So you see that these are 3-month-old organoids. I already have a good expression of PSD95. And then on top is my control, and below is the patient. This is for our nonsense mutation. So we see that in the patient, we have a much lower expression of PSD95. Then MAP2, that's a marker for dendrites and neurites in general. And so we do that one, and then DAPI is just for nuclear staining. This is our merged image. And now I want you to imagine that this PSD95 is about, this puncta that we're looking at is about 0.3 micrometers in diameter. So it's very, very tiny. And this slice that we're looking at, it's 20 micrometers thick. And so this image, at the end of the day, is very messy, and we can't quantify this manually. So it takes a process for us to run this through an algorithm and actually create this surface mask here. So what we see here, this is a mask that we generated for the MAP2 positive cells. So these are basically the dendrites that we can account for. Below, we have a mask for the PSD95, so these are individual puncta, and then on top we have one overlapping with the other. So what we want to take into consideration here is the overlap of PSD95 when in contact with MAP2, and that's what we are going to be quantifying. So far, this is what we have seen. So for all the darker bar here, that would be our controls, and then the lighter bar, that would be the patient, right? So we have our 4 different mutations here, and as you can see, for all of our mutations, we see an underexpression, a lower expression of PSD95 compared to control, except for the splice site mutation. In the splice site mutation, for some reason, we are seeing an overexpression of PSD. Now, this is probably not the functional full-length version of the protein, but it shows that even a truncated isoform of this protein is still traveling to the dendrites. And that is an important notice that we should probably discuss later. Now, this is another analysis, and please bear with me. This is very preliminary data. I received this week as well, so I only want to discuss it very lightly, but I'm super excited to take a deeper look into this later on. So I know these look a little confusing, but I will try to break them down for you. Here we have what we call heat maps, and so I have one heat map of the splice mutation on my right, and then I have a heat map of the nonsense mutation on my left. And because we're seeing different expressions of the PSD95 for these two, I thought it would be interesting to compare both of them. This is a bulk RNA sequencing analysis. So what we do is that we grab 10 organoids from each of the lines. We extract only the RNA from these samples, and we send them for sequencing. We see what kind of transcripts are in that RNA. Right? And then we take a look. So this is going to give me a very broad profiling of what is going on in these samples and try to give me an idea of how these mutations might be affecting downstream pathways of these neurons.
So these are RNAs from all thousands of different genes, not just from the DLG4 gene?
No. Yeah.
Okay.
Yes.
Yeah.
So in here, we're trying to understand what kind of downstream modifications would this mutation cause in this organoid, right? And so in here, we already see a couple of interesting evidence. It's very preliminary, not a lot of conclusions, but we already see a couple interesting things. So these are only the top 50 most differentially expressed. So these are the top differences between control and mutant. And what we see here on the left, for example, we see that there is one protein called NeuroD2. Right here, it's in the green arrow. So NeuroD2, we see that it is it's downregulated in the nonsense mutation compared to the control. However, on the splice site mutation, we see that NeuroD2 is actually upregulated. And so there's something going on here that is really changing the overall profile of these two different types of organoids, right? And then we see that for some other genes as well. We have the Zik family, which is important for neurodevelopment as well.
On your Vanna white.
Yeah. Sorry. Thank you. Yeah, on the green arrow, on the blue arrow, yeah. And then, interestingly enough as well, we have that same marker that I mentioned in the beginning, which is called TBR1. TBR1 is a marker, again, for glutamatergic neurons. And it's also downregulated in the nonsense mutations, but we don't see it on the splice site. But this is just some very preliminary data that we still have to look deeper into. But I thought it would be interesting to mention that it's definitely generating some bigger modifications on the overall scenario of these organoids. Whoop. Yes, now finally, we have a lot of next steps up to come that I'll be very excited to present them on the next conference next year. And yeah, so the first thing we want to do is confirm these truncated isoforms through an RT-PCR, right? And definitely run some Western blots too, see the abundance of these proteins and all of that. Second thing we need to do is finish this transcriptomic profiling through the RNA sequencing, right? We want to really uncover the downstream pathways affected by the PSD95 disruption. And then a third thing, which I was really excited to see that has already been done, or started to be done, is the MEA recordings to do some functional characterization via electrophysiology of these organoids, right? I'm sure we will find something. If the organoids, or if our model is old enough to express PSD95 and to start expressing the symptoms of the mutation, I'm sure we will see differences in neuronal network and even some seizures. Yeah. So this is what I have for today. Thank you very much, everyone, for allowing me to do this today. This has been my first conference, so I'm very excited to be here. Yes, and yes.
Go to the previous slide. It's just so I can be clear here. So you're getting, this is one mutation. This is one mutation here,
Yeah,
and this is another mutation
yes,
there.
sorry, I didn't explain. So over here.
Yeah.
Over here we have the legend.
That's,
so,
yeah. Okay.
Yeah. So what's in blue,
Yes.
the first blue bar,
Yes.
that would be our control. And then the pink bar, that would be the mutation.
Oh, so this is normal,
Yes.
normal, and these are the actual mutations.
Thanks for explaining. Yeah.
No, that's fine. But then there's dramatically different. If you look at,
There's dramatically different changes. I mean, the
Yes.
list of genes changes as well,
Yes.
which is why I'm trying to point out to specific genes that did show up
Yes.
in the top 50 most differentially expressed,
Right.
which I thought was very different. So we
Yes.
have several genes from this Zik family.
Right.
Then we have NeuroD2. We have a lot of genes related to the embryonic development and neurodevelopment of these.
Okay,
Yeah.
okay.
Which are there. I'm sure some of the researchers here will identify some of the genes.
Okay.
And yeah,
wow,
so that's
much to
what we
work
have
with.
for today.
Very impressive.
Thank you so much.
What's with everybody just doing all the work in the last 2 weeks? You should have conferences every month or two. And then